The announcing of the Nobel Prizes is an exciting time of year. These awards acknowledge not only great scientists and science, but also our value and appreciation of new knowledge. Some years, the prizes award discoveries about new phenomena that maybe even a few years previous we wouldn't have been able to imagine. And sometimes the prizes award discoveries about phenomena long appreciated but challenging to investigate and incompletely understood. This year's award in physiology or medicine is of this latter type and recognizes advances in how we sense the world around us and inside us. We have very specialized sense organs that give us the ability to hear, see, smell, and taste, and finally touch. The molecular workings of all of our senses has long perplexed scientists, but our sense of touch has been particularly tricky to study. Unlike the other senses, our sense of touch is of course not limited to our fingers and our skin, like this figure may suggest. The diverse organs and roles of our sense of touch has challenged the identification of molecular receptors for the sense of touch. This year's Nobel Prize in Physiology or Medicine goes to two scientists, David Julius and Ardem Patapoutian, who advanced our understanding of our sense of touch, and in particular, the identification of the molecular sensory receptors for temperature and touch. David Julius is a researcher at the University of California in San Francisco. Ardem Patapoutian was originally born in Lebanon, but emigrated to the United States and now works at the Scripps Research Institute in San Diego, California. Before jumping into their discoveries, let's take a moment to appreciate our sense of touch, which is at the same time both remarkable and underappreciated. Imagine taking a walk on the beach. You can feel the warm sun against your face. You can feel the sand under your feet. Maybe you even feel the pain of a sharp shell you step on. You might feel the tug of the leash as your dog walks ahead of you. And even just walking requires your brain to know the position of your legs and arms. Each of these phenomena requires your sense of touch, and specifically your sense of temperature, to sense the warmth of the sun, for example, and pressure, to sense the sand beneath your feet, or the tug of the leash, or even where your legs are so you can walk across the beach. From this example, we can appreciate that there are sensory receptors throughout our body, and these receptors give us the ability to feel temperature, to feel pain, and to feel pressure, which we need for our sense of touch, but also our sense of self-movement. And these receptors are not only on the surface of the body to sense the outside world, but are also inside our body to monitor internal processes. But how is this remarkable feat accomplished? Now, many philosophers, scientists, and just about every child has thought about how our sense of touch works. One mechanism was proposed by the famous philosopher, mathematician, and scientist René Descartes. In his Treatise of Man, Descartes suggested a very mechanical model in which a series of threads connected the skin to the brain. Physical stimuli, like the heat of the fire shown in this illustration, would tug on these threads and open valves in the brain to allow the influx of so-called humors that led to sensory perception. Now, although many aspects of this model are not scientifically correct, Descartes importantly and correctly pointed out that our senses are essential for really our sense of reality, our connectedness with the world around us, and what he may not have fully appreciated inside us. Since Descartes, many scientists have contributed greatly to our understanding of how signals are sent to the brain. The threads that Descartes imagined are in fact nerve cells, and specialized nerve cells are found throughout the body and relay information about temperature, pain, and pressure, and other information to the spinal cord and the brain using electrical and not so much mechanical signals. But an important and very fundamental question remained. How were individual sensory nerves able to convert physical stimuli like temperature and pressure into electrical signals? And more specifically, what are the molecular receptors present on nerve cells? Now, these discoveries are the basis of this year's Nobel Prize in Physiology or Medicine. To discover the receptors for temperature and pain, David Julius took advantage of a rather unconventional tool, chili peppers. Chili peppers give a warm spiciness to foods, but in some cases, this spiciness is a bit too much and can even be painful. Now, it was known that capsaicin was the active molecule in chili peppers and that it somehow activated sensory nerves to cause the sensation of warmth and even pain. Now, I should add that these receptors for capsaicin are not only in the mouth, but also throughout the body. And if you've ever had the misfortune of cooking with spicy chili peppers without gloves, or worse yet, then touching your eyes, you can appreciate from the burning on your skin or in your eyes that these capsaicin receptors are present throughout our body. To discover the molecular receptor for capsaicin, and thus the molecular receptor for temperature and pain, David Julius's group started with a cell line that was known not to be sensitive to capsaicin. His group then, one by one, expressed candidate receptors in these cells from a list of approximately 16,000 candidates to see which one then endowed these cells with sensitivity to capsaicin. With this approach, his group identified the TRPV1 receptor, which is short for Transient Receptor Potential Cation Channel Subfamily V Member 1. This work was published in 1997 in the very prestigious journal Nature. So how exactly does this receptor TRPV1 convert the presence of capsaicin into an electrical signal? It turns out that TRPV1 receptors present in sensory nerves belong to a very special class of molecules called ion channels. These ion channels are present in the cell membrane that separates the inside and outside of the sensory nerve. When capsaicin or heat or painful stimuli are present, they cause these ion channels to open. Opening allows positively charged ions like calcium or sodium to enter the cell. This influx of ions is the electrical stimulation that then triggers relay of the sensory information to the spinal cord and brain. There are also other temperature receptors. A few years later, David Julius and Ardem Patapoutian, our other Nobel laureate, independently discovered the menthol receptor. We find menthol in mint, which gives things like gum and toothpaste their cool sensation. Together, these and other receptors enable encoding of temperature over a range of temperatures from cool to painfully hot. In addition to his contributions to discovering the so-called menthol receptor, Ardem Patapoutian received the Nobel Prize in recognition of his group's work identifying so-called pressure receptors. These receptors were known to be present in sensory nerves and give rise to the perception of touch and self-movement. To identify the pressure receptor, Ardem Patapoutian's group took a similar and yet different approach from that taken by David Julius's group to discover the TRPV1 receptor. Ardem Patapoutian's group started with cells that were known to be sensitive to pressure. His group then, one by one, eliminated expression of a prioritized list of 73 candidate receptors from these cells to see which one, when deleted, caused the loss of pressure sensitivity from these cells. And as the story goes, his group ran through almost the entire list of candidates until candidate number 72 was identified as the pressure receptor and called piezo1 after the Greek for pressure or push. Shortly thereafter, based on the structure of piezo1, a second related pressure receptor, piezo2, was discovered. This work was published in 2010 in the prestigious journal Science. Like TRPV1, piezo1 and 2 are also ion channels. They open in response to the membrane stretch that occurs due to pressure. Opening of piezo1 and 2 ion channels allows positively charged ions, like calcium and sodium, to enter the cell. And again, it's this influx of ions that causes the electrical stimulation that triggers relay of sensory information to the spinal cord and brain. These molecular receptors, TRPV1 and piezo1 and 2, are important in many processes. For example, thanks to work from David Julius's group, we have improved understanding of how temperature sensation regulates body temperature. And that pain sensation is important not only to alert us to bodily injury, but also evolved in neuropathic and visceral pain perception. Thanks to research triggered by Ardem Patapoutian's group, we now appreciate the role of pressure-sensitive sensors in diverse processes, from touch, movement, urination, breathing, and even bone building. Of course, in science, answering one question always triggers new questions, and there are many new and exciting avenues of research ahead triggered by the work of these two Nobel laureates. One very interesting fundamental research question being investigated is how exactly physical stimuli, like temperature, pain, and pressure, cause these sensory receptors, TRPV1 and piezo1 and 2, to open. This work requires collaboration of structural biologists, physiologists, and neuroscientists, among others. Another more translational area of research is using these sensory receptors to identify drugs that can modulate their activity and treat disease. Given the diverse roles of these sensory receptors, the potential impact of this work is enormous and has the power to yield treatments for neuropathic pain to bone remodeling disorders. I want to thank you for this opportunity to share with you the excitement and also promise of this year's Nobel Prize in Physiology or Medicine. I took a lot of inspiration for this talk from resources available at the official Nobel Prize website. You can see the link for that here. And there are also several good reviews of this work, and I point out one here that you can read if you're interested in learning more. Finally, I want to add that this year's Nobel Prize in Physiology or Medicine is seminal in another way. Previous Nobel Prizes have recognized advances in the other sensory systems, including hearing, vision, and smell, which is very closely related to taste. I encourage you to read more about their work via the official Nobel Prize website. So with this year's work, we have now added an important new piece to our understanding of sensory biology, with Nobel Prizes now awarded across all 5 sensory systems. Thank you.